Welcome back, students. Today we are going to discuss some previous year questions related to Unit 9, that is application of biology. So let's discuss some questions. First question: in Swiss cheese, big holes are made by bacterium producing methane gas, machine, fungus releasing a lots of gas while its metabolic activity, or bacterium producing large quantities of carbon dioxide. Okay, so these are the choices. So you just take 5 to 10 second time, then we will discuss the right answer. Swiss cheese, big holes are made by because of the methane gas, or because of the carbon dioxide gas, or because of some metabolic activity, or via machine. There are 4 different options they have given in this particular question. Okay, now let's discuss the correct answer. In Swiss cheese, big hole are made by carbon dioxide because of the production of carbon dioxide. Okay, so this big hole which is present in the Swiss cheese is because of the presence of carbon dioxide which is produced by some bacterial activity. Okay, so correct answer is choice number D. This is not because of the machine, not because of the methane gas, and not because of the metabolic activity. Okay, so correct answer is choice number D. Now, next question, there are 2 statements given in this question, statement 1 and statement 2. Okay, so in the light of the above statement, we have to choose the correct answer out of these 4. So what is the first statement? In first statement, it is mentioned that Trichoderma species is used as a biological control agent against plant pathogen. Okay, and statement 2, ladybird beetle is used for controlling aphids in mustard plant. Okay, so this question is related to something biological control part. Okay, they have given us 2 different statement, and if you studied that particular part of application of biology in very focused way, then you will definitely get the answer. So first option, they have given us the choice, both statement are true, 1 and 2 statement. In second choice, both statement 1 and 2 are false. In third, statement 1 is true, but second is false. Fourth one, statement 1 is false, but second one is true. So out of these 4 choices, we have to analyze which one is the correct. Okay, you just take 5 to 10 second time, and then after that we will discuss the correct answer. Statement 1, Trichoderma species is used as a biological control against plant pathogen. And 2, ladybird beetle is used for controlling aphids in mustard. Okay, now time up. Let's discuss the answer. So you know this, that ladybird beetle, this is this already we have read this about this beetle. Okay, this act as a predator for the aphids. Okay, all the insects which act as a prey for this particular mustard plant. This statement is true. Okay, as a biological control agent, ladybird beetle. And also Trichoderma species is used as a biological control for certain plant pathogen. Both statements are true. So choice number 1 would be the correct answer. Okay, next question, question number 3. The process of using a natural predator to control a pathogen is called. If we use a natural predator to control a prey or pathogen, this particular what is the correct terminology for the same, whether it comes under genetic engineering or biological control or pathogen control or lineament. Out of these four options, which one is the correct answer? Genetic engineering, biological control, pathogen control, or lineament? Yes, you are right. Natural control of predator. Okay, natural predator control too for a prey or pathogen comes under biological control category. Okay, in which natural predator is used by the farmer to just reduce the population of prey. Okay, so what is the correct answer? Biological control would be the correct answer. Next question, question number 4. What is the name of fertilizer prepared from rhizobium culture? Okay, biofertilizer, azotobacterium, rhizobium, or nitrogen? What is the name of fertilizer prepared from rhizobium culture? If we prepare a kind of fertilizer from a bacterial culture, what is the correct term for the same? Yes, it is biofertilizer. What is the right term? Biofertilizer. Okay, azotobacterium is not a biofertilizer. Rhizobium, a type of bacteria. Okay, so biofertilizer, certain kind of bacterial species are there. Okay, so biofertilizer, if we use a kind of bacteria or colosseum of a bacteria or consortia of bacterium, different kind of bacterial strain for improving the quality of soil. Okay, mainly nitrogen content. That time we can use the term biofertilizer for the same. Clear? So correct answer would be the choice number 1. Next, question number 5. A sewage treatment plant or sewage treatment process in which a portion of a decomposer bacteria. So decomposer bacteria, a portion. Okay, a portion of decomposer bacteria present in the waste is called primary treatment, cyclic treatment, activated sludge treatment, or tertiary treatment. There are few steps in sewage treatment process or sewage treatment plant. Okay, so out of these 4 choices, in which treatment plant a portion of decomposer bacteria has been used? You have to analyze that treatment. And what is the right term for the same? Take your time, 5 to 10 seconds. Choice number 1, choice number 2, choice number 3, or choice number 4. Okay, so now let's discuss the right answer. So, a sewage treatment process in which a portion of activated or decomposer bacteria present in the waste is called activated sludge treatment. This is known as activated sludge treatment, and where this portion has been used in the secondary treatment. Okay, not in the primary treatment, because in primary treatment just separation of big particle debris occur. Okay, bacterial decomposition occur in the secondary treatment, and that particular inoculum is known as activated sludge. So right answer would be the choice number 3. Clear? Question number 6, list number 1 and list number 2. They have mentioned so many type of bacterial or viral or fungal strain name in list number 1, and their product has been given in list number 2. We have to match the correct answer. Okay, so A choice Saccharomyces cerevisiae, B choice nucleopolyhedrovirus, then Monoscus purpuris, then Trichoderma polysporum. 4 options they have given in list number 1. And list number 2, their products like cyclosporin A, statin, ethanol, and integrated pest management. We have to choose the correct answer based on the option below 1, 2, 3, and 4. So quickly just take your time just analyze which bacteria, fungus, or virus strain is related to which type of product or process. Saccharomyces cerevisiae is related to this cyclosporin, statin, ethanol, or IPM. Similarly, this virus, similarly for this Monoscus purpureus and Trichoderma polysporum. Okay, now let's discuss the answer. So, Saccharomyces cerevisiae, this is related to the production of ethanol. This is a very common and very known example. Saccharomyces cerevisiae with ethanol. Okay, so A comes with the ethanol. Okay, second is nucleopolyhedrovirus, NPL. Okay, nucleopolyhedrovirus. This is NPV. This is related to integrated pest management. Why? Because this virus is a specific virus that actually attack or infected a type of caterpillar. And this one is very safe for human, wildlife, and other type of species, right? So this NPV virus and this particular virus, NPV virus, is naturally present in the Australia. Okay, so it doesn't show any kind of harm to other species. So it is associated with integrated pest management. Okay, so this B choice comes with the fourth option. Okay, B choice comes with the fourth option. Now, Monoscus purpureus. It is a very good example of what? Monoscus purpureus. It is a main source for which this type of drug, cyclosporin A, has been isolated, separated out. So, C comes with the choice number 1. Okay, and Trichoderma polysporum is mainly associated with for this statin. So, D comes over here, and statin is mainly used for reducing the cholesterol level in the blood. Okay, so this is how we can resemble the correct choice of list 1 to list 2. So now we can analyze A with 3. A with 3, so out of 3 and 4, any one choice. B with 4, B with 4. Okay, so out of 3 and 4, only here B comes with the 4. So choice number 3 would be the correct answer. C with the choice number 1 correct, and D with 2 correct. So 3 choice would be the correct answer of this question. Clear? So in so many competitive examination, they can given the list of various bacteria, virus, or fungus strain and their products. So we have to remember the product name as well as the bacterial strain name. Okay. Now question number 7, an important application of biotechnology in the field of agriculture. Okay, application of biotechnology in the field of agriculture is to increase the use of nitrogen, phosphorus, and the soil, or increase the vigor of pest and nematodes, or produce pest resistance variety of plant, or cause stress in plants. An important application of biotechnology. So, with the help of biotechnology, out of these choices, you know this, with the help of biotechnology or tool of molecular biology, one can develop a pest resistance variety. Okay, pest resistant variety. So, what is the correct answer for the same? Choice number 3. Because other options like cause stress in plant with the help of biotechnology, why scientists generate a kind of stress to the plant. Okay, and increase use of nitrogen, phosphorus, and sulfur. This is again improve the salinity of the soil. So this is again is not a type of application of biotechnology. Similarly for the second choice. So with the help of biotechnology, scientists develop pest resistant variety of the plant. Also stress resistance variety or any kind of variety which can improve their crop type or product type under harsh conditions. Okay, so choice number 3 would be the correct answer of this question. Next question, question number 8. During which stage of the purification of sewage water are microbe used? At which stage we microbe use? During which stage? We have just now 3-4 questions back we use discuss this kind of questions. So at which stage? Or at primary stage, or secondary treatment, or tertiary treatment, or both 1 and 2? If you learn that particular diagram, okay, then you can very easily give the answer. During which stage of the purification of sewage water are microbe used? Yes, during secondary treatment. At this time, that inoculum of bacterial and fungal strain is actually used to decompose the organic matter present in the sewage water. Okay, in primary treatment, just separation of debris occur, and in tertiary treatment, treatment with the UV lights, chlorine occur. Okay, so mainly in secondary treatment, use of microbe or inoculum has been used. Okay, now next question, question number 9. What does high value of BOD indicate? If BOD is high, then what does it mean? BOD means biochemical oxygen demand. If BOD is high, is the meaning is that water is pure, or that water is less polluted, or that water is highly polluted, or that consumption of organic matter by microbes is higher in the water. What is the meaning of high value of BOD? Biochemical oxygen demand. You just take 5 to 10 second time. Choice number A, B, C, or D. Okay, so if BOD is high, means too many bacteria are present, activated bacteria are present in that sample, water sample. And why they are so much active? Because there are so many organic compound is present, means water is highly polluted. And for the decomposition purpose, they require more oxygen. Okay, so that kind of water is highly polluted one. So correct answer would be the choice number C. Clear? Next question, question number 10. For the production of ethanol, the most common substrate used in distilleries is soya meal, molasses, ground gram, or corn meal. It is a very common question and very good question also. For the production of ethanol, most common substrate used in the industries or distilleries, soya meal, molasses, ground gram, or corn meal. Yes, molasses. Molasses is the right answer. Okay. Now, question number 11. This is not a biofertilizer. Not a biofertilizer. Agrobacterium, non-stalk, rhizobium, mycorrhiza. Not a biofertilizer. You just analyze all kind of bacteria or fungus and plant interaction. This is not a biofertilizer. Okay. So, non-stalk is a nitrogen fixing bacteria. Rhizobium again a symbiotic relationship with the root nodules of pulses. Mycorrhiza again fungal and plant association, right? So again act as a biofertilizer. But this agrobacterium, this actually act as a transfer material of some genes from one plant to another plant. So this agrobacterium is not act as a biofertilizer, not at all. While other three as a free living or in association with the plant species with bacteria or fungus act as a biofertilizer. So out of these four choices, agrobacterium is a kind of choice which or kind of bacteria which is not used as a biofertilizer purpose. Okay, it is used as in a transformation experiments. Okay, now question number 12. They have given us two different statement number 1 and 2, and again they have asked to choose the correct answer. So first I'll read the statement they have given. Statement number 1: plant breeding aim to improve the characteristic of plants so that they become more desirable agronomically and economically. Yes, this is a correct statement. Now, statement number 2: it is a quick and easy method for manipulating plant genetically. Okay, so we have to choose the correct answer. Whether both statements are true or both are false. One is true, second is false, one is false, second is true. They have mentioned in second statement it is a quick process, plant breeding method, and easy method. If we want to manipulate some genetic modification in a plant species, it is a quick and easy method. They have mentioned this in statement 2. Is it so? Just take 5 to 10 second time. Okay. Now let's discuss statement 1. Plant breeding aim to improve the characteristic of plants. Yes. So that they become more desirable agronomically and economically. Yes, this is absolutely correct statement. But this is not a quick and easy method for manipulating plant genetically. It will take time. This is not a quick and easy method. Okay. So first statement is correct, but second is false. So according to me, first statement is true, but second is false. Choice number 3 would be the correct answer. Next, the bacteria used in biogas plant are methanogen, azotobacter, pathogenic, actinomyces. Bacteria used in biogas production. It's a very easy question. Just take your 5-second time and then tell me the correct answer in the chat box. Bacteria used in biogas plants are known as methanogens. What is the correct name of those type of bacteria? Methanogens. Methane producing bacteria. Okay. So not azotobacter. This is a nitrogen fixing bacteria. Pathogenic, no. Actinomycetes, fungus, no. Methanogens. Okay. Choice number A would be the correct answer. Next question, the Green Revolution. Out of these three famous scientists, what is the name of that particular scientist which is mainly known for Green Revolution? Robert Watson, M.S. Swaminathan, Rachel Carson, or none of the above? Yes, you are right. M.S. Swaminathan would be the correct choice. Clear? Now, next question, cross breeding between different breeds. Cross breeding between different breeds are known as line breeding, consanguinity, outcrossing, or none of the above. Line breeding, consanguinity, outcrossing, or none of the above. Cross breeding between different breeds is known as. Choice number A, B, C, or D. Yes. Okay. So line breeding is a kind of inbreeding. Okay. Just to obtain a pure line. So this is not the different breed. Consanguinity means uh, mating between the closely related individuals of the same breed. Again, a same kind of things, same common ancestor. Okay, but different breeds means different crossbreeding between different breeds is known as outcrossing. What is the right term for the same? Outcrossing. Okay, breeding between different breeds. Okay, this what is the right term? Crossbreeding. So and D, none of the above. So right answer would be the choice number 3. Question number 16. Symbiotic nitrogen fixation is carried out by symbiotic relationship is carried out by Bacillus subtilis, Rhizobium leguminosarum, Azotobacter, Xanthomonas citrei. So it is a very common question and very famous question also. As you know, this symbiotic relationship occur between the root nodules of the leguminous plant and Rhizobium bacteria. Okay, others are free living bacteria. Okay, so Rhizobium leguminosarum shows symbiotic relationship with the pulses or leguminous plant. Okay, so symbiotic nitrogen fixation is carried out by Rhizobium. Choice number B would be the correct answer. Next, question number 17. Correct sequence of steps in tissue culture techniques. Correct sequence. We have to analyze the or put all these statement in a correct statement. Okay. Correct sequence of steps in tissue culture techniques. A. Selection of explant. Okay. B. Subculture on rooting and shooting media. Then surface sterilization of the explant. Then acclimatization. Then culturing the explant on MS basal media supplemented with some PGPRs or plant growth regulators. Okay, choose the correct answer from the option given below 1, 2, 3, 4. Okay, so you just quickly put all the option in a correct sequence. What would be the first step? First step is selection of explant. We have to select the explant. This one must be put on the first position, right? And then we have surface sterilize that explant, right? Before putting in any type of media. So this is the second statement, right? And then culturing the explant on MS vessel media for their growth, right? With so this must be at the third position. And after that we have put um, some specific hormones in the media, and then again subculture that explant into the dead media, and that hormone is related to shooting and rooting purpose. Okay, initially to growth, callus formation, and then after that rooting and shooting, second step. So first is selection of explant, then sterilization of the explant, then put that explant on a MS basal media, and then subculture that explant on rooting and shooting media. So this is the fourth step. And after the rooting occur, after some time, just put that explant outside the media. Okay, or in that particular jar with some holes on the plastic for oxygen and carbon dioxide exchange. So stomata also develop and mature enough, and then put the plant on the outside of that particular jar, and this process is known as acclimatization. So this must be the last step, acclimatization. So out of these four choices, D is put on the last, only on the second choice. So this must be the correct answer. ACEPD. Okay, clear? Acclimatization must be the right, clear, and selection of explant must be at the first position. Clear? So this is how you can also analyze out of these four choices which one is the correct sequence. Next, question number 18. Find out the organism which is a source of SCP, single cell protein. Okay, single cell protein, spirulina, paramecium, mushroom, or puffers. Spirulina, paramecium, mushroom, or buffers. Organism which is a good source of single cell protein. Yes, paramecium, wrong. Mushroom, again, this is not a single cell protein. Buffer, absolutely wrong. Spirulina, a kind of algae. This particular organ is considered as a very good source of single cell protein. So choice number 1 would be the correct answer. Next, somatic hybridization is achieved through somatic hybridization is achieved through polymerization, conjugation, RNA mutation technology, or protoplasm fusion. First choice, second choice, third choice, or fourth choice. Just take 5 to 10 second time. Quickly tell me the answer. Polymerization. Is it polymerization? No. This is something different. Conjugation. Fusion of two bacterial cell. No. Somatic hybridization. RNA. Mutation technology. A kind of mutation occur in this technology? No. Somatic hybridization. Fusion of protoplasm. Just removal of cell wall, right, of two cells, and fusion of protoplasm that containing two nucleus of both the cells, right? So fusion of protoplast fusion is known as somatic hybridization. This would be the correct answer. Okay, with the help of this protoplast fusion, tomato is a very good example, which we have discussed it in the literature time also. Okay, so this is known as somatic hybridization. Somatic hybridization is achieved through protoplast fusion. Clear? Now, the last question of this particular video lecture series, callus. Callus is produced by the division of explant, sapling, embryo, or clone. This is related to plant tissue culture. So, callus is produced by the division of embryo, clone. No, it is produced by the division of explant. Explant means any piece of a small particle of that particular plant, maybe leaf or something. Okay, so abnormal growth or multi-growth is there. If we put that particular leaf in a nutrient media, then they can develop a cell of mass, okay, known as callus. Okay, so callus is produced by the division of explant. Explant would be the correct answer. Okay, so this is all question. This is some questions related to some part of application of biology, means part 1, part 2, and part 3. Some more question we will be discussing in upcoming lecture. If you have any doubt related to these questions, please let me know and keep solving some more questions related to these topics. Okay, till then, take care, bye bye, and keep learning.